took his undergraduate work and his graduate work in Canada, the University of British Columbia, so we have recruited a Canadian. He did his postdoctoral work in Cambridge, England, and he was then recruited to the University of Irvine in 2000. And Hans was featured on 60 Minutes 3 years ago. He's managed to survive that. But at that time, he was interviewed widely, and one of the things that Hans mentioned at that time that I thought was interesting was that he decided that he wanted to focus on spinal cord injury at a pretty young age. So he has, for most of his formative science years, really thought about this condition and what it's going to take to really provide, if not total cures, certainly amelioration of the condition. So it's with a great deal of pleasure that I introduce you to Dr. Hans Kierstead, who is co-director now of the Stem Cell Research Center and a member of the Reeve Center faculty at the University of California, Irvine. Hans?
Thanks a lot, Anne. Real pleasure to be here. I think these types of meetings are growing more and more crucial as we start to see things that are coming up from the literature, from the laboratories across the world, and look to be translatable. So it's really important that we have these discussions. I'm going to speak about a couple of the two most recent treatments that I've been working on, potential treatments for spinal cord injury that involve stem cells. And the idea here is to shortcut this natural process where this poor lonely sperm has to make it across a terrific terrain to this oocyte. And we take advantage of fertility clinics in the nation to provide us with the 3 to 5 day old union of sperm and egg and procure blastocysts. And these blastocysts are really marvelous, marvelous things with a great deal of mystery wrapped up in them. And we lose a lot of that mystery when we break open this tennis ball and take the embryonic stem cells outside. This tennis ball is really the thing that tells these cells when to divide and what to become in order. And we lose that instruction manual, and we gain a cell population that's very, very naive. It has the property of reproducing itself almost ad infinitum. When I first started looking into a commercial and clinically viable population of cells, I turned to embryonic stem cells because they're the only cell type, I believe, right now that one can generate virtually unlimited quantities of. And if you can't have the commercial viability, you really can't have the clinical viability. So large mass production is important, hence embryonic stem cells. I don't think it's going to be the end of the story, as virtually indefinite propagation of adult cells will, I think, come. It's a technological challenge. Well, when that day comes, we will welcome it, as we're going to see a lot of advantages and disadvantages of different stem cell lines. So this is not a human embryonic stem cell party. It's just that it's the only cell type that satisfies this criterion at the current time. Bulk production, because you people are big and there's lots of you. We've begun to derive new stem cell lines at UCI in the Stem Cell Research Center that I share the directorship with Peter Donovan. And we set up an embryology lab quite some time ago, and I have secured quite a large source of oocytes and blastocysts from California fertility clinics. So we've been able to derive lines from morula, blastocysts, and epiblasts in a completely clinically compliant manner. So these things are born and bred for the clinic, basically. They've never seen any rodent reagents, no matrigel, fully defined media. And we can grow these things up, and we decided to adopt the nomenclature of human, small h, and then ESC to HUCI. We realized it was called the Hoochie lines, which I think we're going to keep because everyone quite likes them. In any case, we've made a number of lines now that we're just fully characterizing, and given our tremendous supply of blastocysts, both genetically normal and genetically abnormal, over the coming year we are going to, after full characterization and 10 and 20 cell passage, karyotyping and methylation, and really bring these things up to a good high quality, release them to the world and give them away for free. So keep your eye on that ball. We're just about to release a few of them, but I'd like the scientific quality of them to be quite high. They seem to grow very, very well. So having made some new lines, of course, the great challenge, in my view, is to derive a high purity population. And you may not want more of this, but we all want more of that. And that's really, in my view, the greatest challenge and the greatest impediment to the stem cell field. That's it, is we need to derive high purity human cell populations that are excellent research tools and therefore, and also excellent therapeutics. So if we had such a thing, if we had high purity human cell populations, which really the world does not possess right now, then we would be able to use those things for cellular replacement strategies and to put in plate programs for drug screening and predictive toxicology. These screening programs are, can really only be approached if one has a very high purity human cell population. You dump drugs on a 96-well plate for predictive tox or screening. It's no good if what's in that plate is a mixture of cell types. It has to be very, very highly defined. Otherwise, screening it's really not amenable to screening. So what I decided to do in my laboratory is really run after this goal here to drive these cells, and with two other stipulations. Without these things, we do not move forward. They have to be FDA compliant, so we are GTP and GMP. GTP is really just good manufacturing for cell culture. And commercial viability and scaling for clinical viability. And again, given that the first HESC-based clinical trial is probably going to be about $300 million, that's not the kind of money that we're going to get from anywhere but industry. And so it is really the role of industry. Industry is not going to get involved if it's not commercially viable. So I'll just tell you quickly two stories that I've been working on. One you've heard about, but I'll just rush over that one and give you some new data. And then a brand new one that I've been working on that's unpublished. And then what I'll do is, having run through those two scientific examples, I'll then concentrate the last third of my talk on just the barriers, so using those things as examples, and what we have learned moving this technology through to the clinic. Both of these approaches are actually quite close to human application, so we've learned a few things along the way, and I'll use them as examples to elucidate and illuminate those. So we have taken neural populations and derived neuronal and then high purity oligodendrocytes or motor neuron populations, both of which of course are potential targets in spinal cord injury. And one would imagine that if one could have high purity human oligodendrocytes or high purity human motor neurons, we may be able to have an incremental advance and betterment in the outcome of spinal cord injury. The first story, the oligodendrocytes, is really predicated on a series of studies that I really jumped on a very long-standing bandwagon. And demyelination has been documented by dozens of researchers over a couple of decades in almost every species. The most poorly documented is humans, because of the limited access to properly perfused tissue that we're trying to address at the Reeve Irvine by setting up a new tissue repository where the tissue is actually post-fixed and handled for resin embedding and electron microscopy. That's going to be quite a unique resource. I'm trying to set that up. I think we've got over the major barriers, so hopefully that resource is going to be available within a year or two. In the white matter of most species, we have dying oligodendrocytes that result in demyelination after spinal cord injury. And I've done a number of papers on this I'm not going to go through, but that I believe justify that demyelination is a therapeutic target. There's not lots of it, but it is there. And so my viewpoint is that it is a bona fide therapeutic target. If we address that target accurately, we'll have an incremental advance in the functional outcome. This is not the type of thing that's going to get people jumping and running around again and playing soccer, because we're not doing anything to fix the cavitation and the majority of tissue that was lost after the injury. It's really a patch job of what's left, getting it to work again. So I developed a 42-day protocol. That's a very long differentiation protocol. It's the longest one I know of. I've never read of another differentiation protocol that is as long as that. I think that alone is one of the reasons for the high purity. And I'm not going to go over the basic science, but we committed these cells to a neural progenitor type by re-engineering the retinoic acid regime. Without that re-engineering, we could not get high purity. And then de novo developing a differentiation protocol for high purity oligodendrocyte development.